first one are the intracellular receptors, and the second one are the cell surface receptor. So if you read these terms carefully, you can get the answer from this thing itself. So intracellular, what do we mean by intracellular? Intra means within the cell, and intracellular means within the cell. So they belong to the nuclear receptor family of the highly conserved transcription factors. They regulate negatively and both positively. The ligands for the same could be small and hydrophobic. Examples for the intracellular ligands could be steroid receptors, estrogen, testosterone, thyroid, vitamin D, sex hormones, orphan receptors, and retinoid. So in very simple explanation, what we have to remember is that intracellular receptor, they belong to the nuclear receptor, first thing. Second thing, they are a family of conserved transcription factors. ये उसका basic nature होता है. And third thing, they regulate the complete functioning both positively and negatively. What do we mean by positively and negatively? So if you have to understand the function of anything as both positive and negative, positive means that presence of that particular thing would positively affect, and negatively means नेगेटिव का मतलब हमेशा नेगेटिव रिजल्ट्स नहीं होता. समटाइम्स absence of something could be positive for a response. So intracellular receptors are they regulate the complete cell signaling both positively and negatively. The second one is about the cell surface receptors. So they are also known as the transmembrane receptor. They are found attached to the plasma membrane. The receptor binds to the extracellular ligand molecule because the ligand do not travel or they do not cross the cell membranes. The cell surface receptor have mainly three domains. The first one is external, second one is the hydrophobic spanning, and the third one is intracellular domain. So give me an example of cell surface receptor according to your understanding.
GPCR.
GPCR. Excellent. Which is the most common example for the cell surface receptor, in which we have this particular plasma membrane, we have this ligand, then we have 1, 2, and 7. So we have this particular function, this particular structure, in which we have first the extracellular ligand binding domain, so this particular domain. Then we have the hydrophobic membrane spanning region. This is the hydrophobic membrane spanning region, and the third one is intracellular domain inside the cell. That means this particular region. The working is basically, first there occurs the dimerization and then there occurs the conformational change. Dimerization means the ligand would come and bind to the receptor. This is the dimerization in simple meaning, and then there occurs conformational change in the complete structure, and finally the work would happen. The categories of the same are, first one is ion linked channel receptors, then we have enzyme linked receptors, and third one are the GPCR receptors. So these are okay. So these are mainly the three ones जो अभी हमने discuss किए. The very first one as the ion channel linked receptors. So they are also with the transmitter. They're also known as the transmitter gated ion channel or the ionotropic receptors. They bind to the ligand and open a channel through the membrane that allows the specific ions to pass through. Simple meaning, we have this particular plasma membrane, and ion channel linked receptor means those receptors whose functioning is basically based upon a specific ion. So whenever we have a specific ion bound to the receptor, this would allow the specific ions to pass through and the complete functioning would occur. So it binds to the ligand and opens a channel through the membrane that allows specific ions to pass through. Now, extensive membrane spanning regions. When a ligand binds to the extracellular region of the channel, there is a conformational change in the protein structure. So that means you have to remember about one thing, that there would occur conformational change in the protein structure. It allows the ions like Na, calcium, magnesium, hydrogen to pass through the multi-pass transmembrane proteins. Examples for this are acetylcholine, that is a neurotransmitter. It allows to act as a ligand on the acetylcholine ion cation channel receptor. Now, there is one thing you have to remember, that is acetylcholine is a cation. First thing, because ये पार्ट सी के अंदर क्वेश्चन आया हुआ है, इन विच मतलब क्वेश्चन बहुत बड़ा था एंड एंड के अंदर जो आंसर का कंक्लूजन था वो उसके केटायन नेचर के ऊपर बेस्ड था. सो रिमेंबर अबाउट दिस. द सेकंड वन इज एंजाइम लिंक्ड रिसेप्टर्स. सो एंजाइम लिंक्ड रिसेप्टर मींस वो रिसेप्टर्स जो एक स्पेसिफिक एंजाइम के ऊपर या फिर उनका फंक्शनिंग, either that means their functioning is based upon a specific enzyme. So the cell surface receptors with the intracellular domain that is associated with the enzyme. The intracellular domain of the receptor is itself is an enzyme. The signal is transferred through the membrane activating the enzyme. Example, the receptor tyrosine kinase, in which the C domain acts as an enzyme, receptor tyrosine associated receptors, and then we have serine threonine kinase receptor. Simple मतलब इस कहानी को समझने से पहले you have a receptor in which the complete structure which we have, in that complete structure जो उसका intracellular domain होगा, we have understood about the extracellular, the membrane spanning region and the intracellular domain. So जो उसका intracellular domain होगा, that actually acts as an enzyme itself. So when we have the ligand, we have the receptor and we have the ligand, then there occurs the signaling. Now because of the binding or because of the dimerization over here, there occurs a conformational change. After conformational change, this particular intracellular domain, which is functioning as an enzyme, it would get activated, and finally the subsequent transduction of the signal would occur. The third one are the